KETA ML Chemiluminescence and Fluorescence Imaging System. Operating Features Overview. To begin, turn on the main power switch on the right side of the unit. Then, press the front activation switch to launch the imaging software, Magic Blot. Once the main software interface appears, click Capture. The camera will begin a 5-minute cooling process before imaging can commence. The KETA MLX2 is a multifunctional imaging system equipped with sample tray automatic recognition. By placing different sample trays, the system can identify and switch between various imaging modes, including chemiluminescence, fluorescence, white light, blue light, and UV imaging. Inserting a white tray automatically activates the chemiluminescence imaging mode. Inserting a black tray automatically activates the fluorescence imaging mode. Inserting a light box automatically activates the gel documentation imaging mode. Once the camera cooling is complete, click Settings to perform a darkroom calibration. Select the calibration mode and click Calibration. Then Start to begin the process. This function is typically used for initial setup to ensure a background free of bright spots. Next, click Settings at the bottom of the main screen and navigate to Imaging to configure imaging settings. First, set the binning mode to either high resolution, normal, high sensitivity, or ultra-high sensitivity. Select Normal. The system provides distortion correction for spherical aberration and flat field correction for uniform illumination. Once configured, imaging can begin. Chemiluminescence imaging mode. Insert the white sample tray to activate the chemiluminescence mode. Place the test sample and use the Live View function to center it properly. Since KETA MLX2 supports fully automated imaging, click Autofocus to focus automatically. On the right panel, there are 3 capture modes: A. Auto Capture, Q. Auto Quant Capture, M. Manual Capture. Select Auto Capture and click Next to capture the chemiluminescent sample. The system will then capture the marker, standard reference. After acquisition, the software automatically merges the sample and marker into a combined image. Click Export to save the image in formats such as TIFF or JPG. You can also click Save to enter the editing interface, where separate images of chemi, marker, and combine can be stored individually. Clicking the printer icon generates a report and exports it as a PDF. Auto Quant Capture. This mode stops imaging before overexposure occurs. The process is the same as auto capture. First, the sample is captured, then the marker, and finally, they are merged into a single image. Next is manual capture, which offers 4 shooting modes: single for single image capture, dynamic for timed acculation, batch for simulating blot imaging, and program, where the user specifies different capture times. First is single capture. Click the time field to set the exposure time in seconds. Then click Capture to take the image. After confirming, click Marker to set the exposure time. Once the capture is complete, the sample image and the marker will be automatically merged into a single image. For dynamic capture, users specify the number of images and time intervals. For example, set to capture 1 image every 10 seconds for a total of 10 images. During the capture process, each image can be viewed in real time. Click the adjustment icon below to modify image settings, such as contrast and brightness. After capture is complete, users can either select all images or choose specific ones for export and saving. The last mode is Batch, which simulates the Western blot exposure mode. After entering the mode, set the capture time and the number of images. Then click Capture. Once the capture process is finished, the images can be exported and saved. Fluorescence imaging mode. In fluorescence capture mode, place the black sample tray to enable automatic fluorescence detection. Click Settings, then select Dyes to edit the preset dyes. Click Insert to add a new dye, enter the dye name, and select the light and filter. Click Save to complete the addition. Before fluorescence imaging, click Live to adjust the sample position and focus. Once adjusted, proceed with fluorescence capture by clicking the plus icon to select the desired dye. After confirmation, begin the capture process. The capture methods are the same as those in chemiluminescence mode, including auto capture, auto quantitative capture, and manual capture. For example, when selecting auto capture, the system will first capture the first dye, then proceed with the second dye. After combining, the image can be exported and saved. Gel documentation imaging mode. This mode supports 3 light sources: blue light, white light, and UV transillumination. Blue light imaging. Select a safe dye such as Cyber Green. Enter capture settings and choose between auto exposure and single exposure. Auto exposure will automatically adjust and capture the image, which can then be exported and saved. For single exposure, manually set the exposure time before capturing the image. White light imaging. Insert the white light box and place the sample inside. Click Live to adjust positioning and focus. Then, proceed with white LED imaging by selecting a dye such as Kumasi Blue. Enter capture settings and choose either auto capture or single capture. Set the exposure time, capture the image, and export, save it. Image editing. To begin editing an image, first select the desired file from Explore. Clicking on the file will open the Edit mode, where you can use the crop function to define the area of interest. After selecting the desired region, confirm the selection to finalize cropping. The software also includes an image revert function. Clicking the restore icon at the bottom will revert the image to its original state. The rotate function allows image rotation in 90-degree increments or at custom angles. Background and presentation. One of the most commonly used functions is background subtraction. Click to enter this mode, select the background color, and confirm to proceed. Within Background Subtract, you can choose Average ROI and drag a selected region to remove the background. Alternatively, using the median filter, you can adjust the kernel size by inputting a value or adjusting the scroll bar. Display control includes invert and color adjust for contrast, brightness, and gamma adjustments. Annotation. The annotation function allows text input as well as drawing lines and shapes. Clicking on text enables users to enter text, adjust font size and color, and apply a background if needed. Measurement. The measurement tool allows users to draw a line to create a ruler, where numerical values, units, font size, and color can be customized. The software also includes a 3D map function. Select a specific region, choose a background color and model color, and the 3D rendering will be displayed on screen. Analysis. The Analyze function includes 1D gel analysis. Begin by selecting a background color and defining lanes. If any lanes are incomplete due to gel damage, additional points can be added for correction. Clicking Detect Lane enables the software to automatically recognize lanes. The Lane Adjust function allows users to delete, add, or modify lanes by adding nodes as needed. Band analysis. Begin with background correction. Selecting either rolling disk or minimum as the correction method. Next, click Detect Band and select All to allow the software to automatically detect bands. Proceed to Lane Adjust, where you can fine-tune individual bands by selecting them from the left panel. This includes deleting unnecessary bands or adding undetected ones. Once adjustments are complete, click Next to proceed to quantification analysis. First, select Molecular Weight Standard for calibration. Click Select MW Stud Lane. Then proceed by clicking Load. The software includes built-in options such as the 100-part billion DNA ladder or lambda DNA, which can be selected for analysis. After selecting the 100 base pair DNA ladder, modifications can be made according to the user's reference standard. Confirm and click Next to generate a report, which can be saved as an Excel file or exported as a PDF. Alternatively, users can select mass standard by choosing a lane or band standard, then loading the corresponding reference. After confirmation, click Next to generate a report, which can also be saved as an Excel file or exported as a PDF. Area density, protein quantification analysis. Start by selecting the background color, then proceed to define region. There are two selection methods, trace and shape. To use automatic tracking, click on the sample with the mouse to select it and confirm. Repeat this process for all samples. Once all samples are selected, click Next to enter Define Background for background subtraction. 3 methods are available: custom range, minimum value, and 0. Alternatively, you can use shape selection, where 2 shape options, rectangle and circle, are provided. Selecting rectangle allows you to copy and apply the background correction to the target samples. After completing this step, proceed to quantification analysis. 2 quantification methods are available. Relative percentage. Select one sample as the 100% reference. Then click Next to calculate the relative percentages of the other samples. A report can be generated and saved as an Excel file or exported as a PDF. User-defined quantification. Input the known molecular weight of specific samples. Example, sample 1 equals 100 kD, sample 5 equals 50 kD. After entering the values, confirm and click Next to calculate the molecular weights of unknown samples. The results can also be saved as an Excel file or exported as a PDF. Colony counting. First, define the colonies by selecting Split Colony or Delete Colony as needed. Then, define the background. For example, using the section fan-shaped method, set 3 regions and select the area for analysis. Click Next to generate a report. Alternatively, select the grid method, choose the target analysis area, and generate the report. Microtiter plate. After entering the module, select the background. Proceed to define region, where 2 methods are available: grid definition and well-shape definition. Using grid definition, select the microtiter plate for analysis, or adjust individual wells by choosing Well Adjust. Then define the background. Next, proceed to quantification, where users can manually input values and select a known concentration. Once confirmed, a report can be generated.